victory, so sweet. Perhaps slightly embellished, but hey, ChatGPT wrote it. I didn't, so you can't blame me. And moreover, I like to think of CRISPR Hal, what we named the machine learning model, more of like a Wall-E rather than Hal, but the name was good, so we stuck with it. Anyway, moving forward, a quick little introduction to CRISPR. Everybody and their mom knows about CRISPR, and it's composed of two components, one of them being a single guide RNA, highlighted here. And what this is it's a nucleic acid sequence that defines where this complex will bind and subsequently cleave. And the second component is the Cas9, and this is the what does the legwork. This does the actual cleaving. So what will happen is, come on, I'm supposed to get some animations here. There it is. So it'll bind over, it'll find an NGG PAM, and then if the upstream sequence is correct, the guide RNA will bind to this, and if it's stable enough, again, I'm having trouble, you'll get a double-stranded break from the Cas enzyme. So because this is very easy to reprogram, what we can do is we can target a bacterial genome, for example. And what'll happen is you'll have the Cas9 with the correct guide RNA. It'll be introduced to the cell that we're targeting. Again, it'll bind the target site. It'll create double-stranded breaks. And unlike mammalian cells, which have very good repair pathways, most bacteria lack these pathways and have trouble resolving these double-stranded breaks. So as a result, they die. Perhaps not that dramatically, but they die nonetheless. So what this allows us to do is to use CRISPR as a strain-specific antibiotic agent that's easily reprogrammable. However, there are a couple drawbacks. Firstly, if you'll look at the X or the Y-axis here, you'll note that we have a varying range of activities and therefore a range of killing efficiencies, going from 0 up to 100%. The second problem on the X-axis is that when we try to predict these guide activities using published algorithms, we have a hard time doing so. So this leads me to the two overall goals of this project. Firstly, to develop a data set that is truly indicative of the Cas9 single guide RNA cleavage landscape in bacteria, and then to use this to train a machine learning model, again, that we call CRISPR Hal, such that we can reliably and accurately predict guide activities. In order to do this, we first need an assay, and we use what's called a dual plasmid assay. So the first plasmid contains Cas9 and a guide RNA, and the guide RNA is targeted towards this PTOX plasmid. And on this PTOX plasmid, there's a toxic gene called CCDB, which we can turn on or off. When we turn it on, cells harboring this plasmid will not grow. So the idea is that we can introduce some plasmids. Each of these cells, let's say, gets one that's red and gets one that's blue. I think that's blue. I am colorblind, so if I'm not right, I apologize. But say the blue one that's targeting PTOX is highly active, you'll get cleavage and subsequent degradation of the PTOX plasmid. In the red example here, it has a perfect match. However, it's not a good guide. It doesn't allow for high cleavage, and therefore the PTOX plasmid remains intact. Therefore, in subsequent outgrowths, when we turn the toxin on, only cells harboring guides which have led to successful cleavage will be able to replicate in the blue scenario here. But on the right, the red one, because the PTOX plasmid was not cleaved and then degraded, it will stop growing. We can then compare what we get in the outcome of this to a non-induced condition or a repressed condition where the toxin was never turned on. So this is what we can compare this to. And the outcome can be read with Illumina by growth curves or percent survival. So this is an example of what the outcome of the assay looks like. In the top row, we have a no guide condition. So the first plasmid has Cas9, but no guide targeting PTOX. So when we put this plasmid into the strain, we see that when PTOX is never turned on, so the toxin's repressed on the left plate here, we get many colonies. But when we turn the toxin on, because there's been no cleavage, PTOX is present, the cells will not grow. Conversely, on the bottom row, we have Cas9 and an active guide RNA. On the left, when the toxin is repressed, we see growth as expected. And on the right, because you're getting cleavage of the toxic plasmid, it's being degraded, and we're seeing growth in these conditions. So we can then calculate the colonies on each of these and take the average or ratio of them and calculate a percent survival. Moving forward, what we did is we took a handful of guides and tested them using this assay. On the left, it's similar to the plates one you saw before. And one thing that you'll note is that you're seeing various percent survival. So it's not just on or just off. We see one here in the middle that we can detect intermediate activity. Furthermore, when we conduct this similar assay, but do an outgrowth in liquid rather than on plates, we see the same thing where the active one is growing well in toxin-inducing conditions. Some are growing okay. And then we see again, no guide is not growing well, similar to the inactive guide. So moving forward, rather than testing these individually like I'd been doing so far, I sought to get a high throughput pooled assay. And the idea is that we picked all the targetable sites on the PTOX plasmid. We ended up with 304 perfectly matching guides. And then we had 2 controls, one of them being 48 non-targeting guides, and then 15 that we call mismatches. So they do have a degree of complementarity, but are not perfectly matching. So the latter 2 shouldn't work as well as the perfectly matching guides. We then tested this pool in a similar manner as I described previously. The idea is that, as mentioned, the ones that are cleaving will grow relative to the rest of the pool, and the ones that aren't cleaving won't. And we should get something which, from the output of Illumina, we can calculate metrics like we see on the right. One of them being the activity, which is a representation of how enriched or depleted the guide was. So we see the blue guide is being cleaved and therefore it's enriched. The red guide is not cleaving and therefore it's depleted. And we can calculate some metrics with which we can estimate cleavage activity. So this is the outcome of that experiment where each dot here represents a single, a unique, I'll call guide from within the pool. The black ones we can see are ones which have been significantly altered from the average, either up or down. The gray ones are perfectly matching guides that are not significant. The red ones are non-targeting and the cyan are mismatch. So firstly, you'll note that the cyan and the red ones are not becoming enriched, which is exactly what we'd expect from negative control. So they're very low on the y-axis, which is good. Secondly, as we saw previously, we can detect a gradation of activity. It's not just on or off. We see a nice kind of continuum of activity along the y-axis. And then furthermore, one thing that's notable is that there doesn't seem to be a strong correlation between abundance and activity. Previous assays will throw the pool in and they'll see what they get out, and they'll assume that guides that are of high abundance or low abundance have a particular activity. But as we see here, a lot of guides are to the right of the 0 on the x-axis, meaning they're high in abundance, however, not necessarily high on the y-axis, meaning high in activity. Conversely, we see some low in abundance that are high in activity. So this assay is able to account for that, which is helpful. So the conclusions we could draw is that abundance does not necessarily equal activity. We can see a range of activities, and furthermore, we can separate active and inactive guides successfully. Moving forward, I wanted to confirm this using a separate assay. So what I did is I tested the guides individually. I plated the pool out. I picked up unique colonies and tested them because I wanted to see if they acted similarly when you test them individually compared to the pool. So to see if the context of the pool kind of changed how we're detecting things. And these are 4 growth curves that I generated. I did about 100 of these, but these are 4 kind of representative ones. First, you see active, so they're growing again well in induced and repressed conditions. Inactive, it grows fine and repressed, which is what we'd expect, but not well in induced, again, because it's not cleaving the target plasmid. But most interestingly is this third category here of toxic guide RNAs. So even when Cas and the toxin are off in the repressed condition, you're seeing poor growth, which is quite curious because it seems that the presence of the guide alone is kind of unfavorable to the cell growth. What we did then is with the 100 or so guides that I calculated these growth curves with, we put them through a program and got the area under the curve as a metric for how well the guides grew within that particular condition. And we plotted that. So that's the plot here on the left, where the x-axis is the area under the curve for induced conditions, and then the y-axis is for repressed conditions. So immediately, first I'll note also the letters, they correspond to what we see in the previous slide here. But we see here the inactive guides, these are not growing in the induced condition, meaning they're not cleaving the toxic plasmid, but they are growing in the repressed condition. So we can cluster these into a group. Conversely, we see ones that are growing well in both conditions, and we'll consider these active. And then we see some that aren't growing well in either condition, and we'll consider these toxic. So I then wanted to map these back to the pooled assay. So these are all the guides you see on the left highlighted from that same plot I showed earlier. And what you can see is that the active ones are clustering high on the y-axis, meaning they're also active in the pooled assay. And then the toxic and inactive ones more or less are clustering low on the y-axis, meaning that it does appear that we successfully separated out active and inactive guides in both these conditions. However, one thing that I will note is that there doesn't seem to be a strong clustering of toxic guides in this, which could highlight that maybe it's just detecting a gradation of toxicity. Some guides clearly are more toxic than others if you look over here, or conversely, just that the pooled assay isn't a valid assay to measure toxicity. Sure, it's good at looking at activity, but maybe not the best for toxicity. And those are the conclusions I could draw from that. So potentially we need to look into an orthogonal assay in order to measure toxicity, but it highlights a neat problem of using this in bacteria, this system. So that's the first part of the project. That's mainly what I did, but I handed the data to the smarter half of the project, Tyler Brown. He works in the Glur lab here at Western as well, and he trained a machine learning model on it. So this is the architecture of the model, and don't worry, because I don't really understand it either, but the idea here is that we have an input sequence, a guide sequence, and we have a known activity from the previous assays that I talked about. And the idea is that pretty much this is just a big algorithm, a big equation, let's say, and they're trying to optimize the input sequence and output activity across all of the guides that we found. What we could then do is pull out a segment of those, and the model never sees those. And we can test how well the model's working by predicting the activity from the model and then comparing it to the known activity from the assay that I was discussing previously. And this is what we see. So the first 4 columns are data and models from previous papers. And you can see they have modest activities or modest correlations between predicted and actual activity. And then on the far right, you'll see CRISPR-Hal. This is the model we created that has a substantially higher correlation between the actual activity and the predicted activity. So from this, I think we achieved both of the project goals that we intended to at the beginning. With that, I'd like to acknowledge everyone that helped me along the way, and I'd like to thank you guys for listening.
Awesome. Thanks so much, Dalton. That was a great talk.
Thank you.
So if anyone has any questions for Dalton, feel free to raise your hand or drop them in the chat. Go ahead, Matt.
That was really cool, Dalton. Thanks for giving the talk.
Thanks, Matt.
I was curious, did you think or look a little bit more into the toxic guides? Like, can you explain that by off-target effects in the genome?
Yeah, so that's like one of the things we're working on next, not from what we have now, but we are planning assays to kind of detect that more accurately. A paper came out recently of toxicity in dCas9. So this is even a problem when you're not seeing cleavage. So there's off-target binding in essential genes that just kind of disrupts the normal growth of the cell. So I'm wondering if what we're seeing here is kind of a pseudo dCas9 activity where it's sufficient homology to an off-target site, not for cleavage, but for kind of just to disrupt the transcription of that essential gene or whatnot. We looked at off-target cleavage, so I don't think that that's it, but I mean, you can never fully count it, I guess, but that's the next step, because I think that these toxic guides are adding some noise to the model such that if we can figure out what's going on with those, we should be able to increase the correlation between predicted and actual activities kind of to their upper limit is what I'm hoping.
For sure. That's cool.
Great. Thanks for the question, Matt.
Awesome. It seems like we have another question in the chat. So did you observe any differences in sgRNA activity in clonal versus pooled growth due to the competitive growth environment in the pooled setting?
Yeah, so just because how we measure the activity is relative abundance within the pooled, that's why we sought to do them individually to kind of compare between them. So more or less, they seem to behave the same between when we detected activity as a pooled versus when we looked at the guides individually. So thankfully, that was the case.
All right. Don't see any more questions. So with that, I think we're going to move on to our next speaker. So Dr. Luge, if you could please share your screen.
Yeah, I'll just share it now.
All right. So I'd just like to welcome Dr. Luge as our next speaker from the University of Manitoba, and she will be discussing uncovering crosstalk between mediators of the unfolded protein response.
Okay, so thank you very much for the opportunity to present today. So I'm going to tell you about the unfolded protein response, what it is, why we should study it, and then towards the end, I'm going to share with you some recent data that we've generated from my group here at the University of Manitoba. So before getting into the specifics of the unfolded protein response, I need to first take you back and remind you of your very fundamental cell biology and reintroduce you to an organelle called the endoplasmic reticulum. So the endoplasmic reticulum is essentially our cell's protein production and packaging plant. So this is where proteins are made on ribosomes that are dotted on the ER surface. And then these newly synthesized proteins are translocated into the ER lumen, where they're folded into their correct shape or conformation before they're shuttled off to the distribution center, which is the Golgi, and then to wherever they're required. So the ER works with absolutely amazing efficiency. And to do this, it requires energy availability. It also requires a calcium-rich environment because many of the ER chaperone proteins that aid in protein folding require calcium binding to function at their optimum capacity. And the ER also requires a highly oxidizing environment to facilitate disulfide bond formation. And when the endoplasmic reticulum has all of these, it's normally meeting the demands placed upon it, and we would say that it's in homeostasis or in balance. But sometimes things do go wrong. So for example, maybe we have a decrease in energy availability, and that's going to impact on the efficiency of protein folding within the ER. Perhaps we've had release of calcium from the ER, and that means that ER chaperone proteins aren't going to work as well. And in both these cases, we start to get a breakdown in ER function. Or alternatively, the ER environment could be maintained, but we're just getting this huge excessive demand being placed upon the ER to fold proteins. And a really good and current example of that is viral infection. So in these instances, essentially the capacity of the ER to meet the demands being placed upon it is overwhelmed, and we start to see the accumulation of unfolded or misfolded proteins. And that's what we mean when we use the terminology endoplasmic reticulum stress. So our cells are constantly on the lookout for ER stress, and they essentially monitor the health of the ER using 3 transmembrane receptors. I'm just going to put my pointer on here, called IRE1, PERK, and ATF6. So these receptors are anchored on the ER membrane, and they have an amino terminus that juts into the ER lumen and a cytosolic C terminus. So in a healthy, unstressed ER, IRE1, PERK, and ATF6 bind to an ER chaperone protein called BIP, which is also known as GRP78. And this binding essentially holds these 3 transmembrane receptors in an off position. But when we get the accumulation of unfolded proteins, which are my pink squiggles here, so this is an ER stress condition, BIP has a higher affinity to bind to these unfolded proteins than it does to IRE1, PERK, or ATF6. So this enables these transmembrane receptors to flip from an off to an on position, and each of these then triggers downstream signaling pathways, and it's these pathways that we collectively refer to as the unfolded protein response. So what does the UPR do? So we can really consider the unfolded protein response as being a little bit like a traffic signal. So we get this general halt in cap-dependent protein translation, and the reason for that is we have an ER that's already stressed, so we don't want to be placing more demands upon it to fold additional protein. Alongside this, we get an increase in the expression of genes encoding ER chaperone proteins and components of the ER-associated degradation machinery. And the idea here is that we refold those proteins that can be refolded, but those that are damaged beyond repair are retrotranslocated out of the ER, and then they're tagged for degradation via the proteasome. And if these two go to plan, we should get the green light again, and ER homeostasis should be restored. So the really important thing to realize is that in healthy cells, the unfolded protein response is only ever meant to be a transient process. We can really consider it as a backup fail-safe mechanism that gives cells a window of opportunity to try and resolve this ER stress and to restore ER homeostasis. But there is going to be certain circumstances where the ER stress is hugely excessive, or maybe it's prolonged. And in these instances, a pro-adaptive or a pro-survival adaptive response isn't sufficient, and cells make a decision to flip from the pro-survival into a pro-death UPR. And in these instances, ER stress-induced cell death ensues and the damaged cell is removed. So this is a schematic of the unfolded protein response, and I'm just going to run through this quickly because some of the data that I'm going to show you later in the talk requires that you understand some of this. So here we have IRE1, and when GRP78, which is also called BIP, dissociates from IRE1, IRE1 dimerizes, and this leads to activation of its kinase domain, so IRE1 trans-autophosphorylates, which facilitates activation of its RNase domain. And the RNase activity of IRE1 has two signaling outputs, XBP1 splicing and RID. So XBP1 splicing is by far the better characterized output. It's classically what we associate with IRE1 RNase activity. So IRE1 excises a 26 nucleotide intron from XBP1, and when this is religated, it's referred to as spliced XBP1. And when it's translated, it forms a potent transcription factor that upregulates the expression of genes encoding ER chaperones and components of the ER-associated degradation machinery. So the other outcome of IRE1 RNase activity is RID, or regulated IRE1 dependent decay. And this is essentially the selective degradation of mRNAs and microRNAs. So RID was first described in 2006 in Drosophila and confirmed in mammalian cells in 2009. So it's, I guess, quite a young biological phenomenon that we're still really trying to get to grips with. Although we don't completely understand RID, we do know that RID signaling could be important in facilitating the flip from a pro-survival to a pro-death UPR response. So now we have PERK, and PERK is very similar to IRE1 in that when GRP78 dissociates, it dimerizes and it trans-autophosphorylates. And the kinase activity of PERK phosphorylates EIF2 alpha on serine 51. And what this does is it basically presses pause on general CAP-dependent protein translation. But this pause is not absolute, as certain genes that have internal ribosome entry sites or upstream open reading frames within their 5' UTRs, such as ATF4 or activating transcription factor 4, are preferentially translated under these conditions. And ATF4 is being shown to increase the expression of genes involved in amino acid biosynthesis, genes which help regulate autophagy, and the proapoptotic factor CHOP. And then finally, we have ATF6. And ATF6 is a little different to IRE1 and PERK in that when GRP78 dissociates from it, ATF6 undergoes this conformational change. And somehow it pops out of the ER and goes to the Golgi. And I'm not sure if I really understand how that occurs yet. And in the Golgi, it's cleaved by site 1 and site 2 proteases, which form the active transcription factor called nuclear ATF6. And ATF6 upregulates genes encoding ER chaperone proteins, but it also upregulates XBP1. And this is interesting because in this way, ATF6 helps support the IRE1 signaling pathway by maintaining a large pool of XBP1 for IRE1 to splice. So as our ability to monitor and track the unfolded protein response has increased, or the mediators of the unfolded protein response, we're starting to realize that it plays a really important role in essential physiological processes. So for example, we know that mediators of the UPR are important in innate immune responses. They've been shown to play roles in learning and memory in the brain. And of course, they're extremely important in beta cells in the pancreas. As we know, beta cells have to make copious amounts of insulin, so they need to have a very robust unfolded protein response. But we're also starting to realize that dysregulated or aberrant signaling via UPR mediators also contributes to the progression of diverse range of diseases, including neurodegenerative diseases such as Alzheimer's, Huntington's, ALS. Aberrant activation of UPR signaling mediators has also been linked to cardiomyocyte cell loss in the heart following stroke or ischemia. We know it plays important roles in inflammatory diseases such as rheumatoid arthritis or Crohn's disease. And one area that is a real hot topic at the moment in the UPR field is diabetes, and understanding how a breakdown in UPR signaling can lead to beta cell loss. But one set of diseases where we know the unfolded protein response mediators play an important role are cancers. So constitutive activation of IRE1, PERK, and to a lesser extent, ATF6 signaling pathways have been reported in diverse cancers ranging from breast cancer, and in particular triple-negative breast cancer, to prostate cancer, pancreatic cancer, lung cancer, brain cancers. We've really reached the stage where if you name a cancer, it is likely that I will be able to find a paper linking one of the UPR mediators to that cancer. So we also know as well that signaling via UPR mediators contributes to all stages of cancer progression, from early stage tumorigenesis right up to later stage metastases and the development of chemoresistance. So it should come as no surprise when I tell you that targeting mediators of the unfolded protein response has been suggested as a potential therapeutic for several cancers. So at the moment, thanks to a lot of work by research labs and companies over the past 10 to 15 years, we do have access to a panel of small molecule inhibitors that can block IRE1, PERK, and ATF6 signaling. So most of the inhibitors developed have been against IRE1, and that's probably a reflection that of these three mediators, IRE1 is the more druggable target. So we have the MKC family, which I'm going to talk about today when I show some of the data, and STF and 4-MeO8C, and these four basically function by directly binding to the RNase domain of IRE1. And then we have Kyra and G030, 89668, just rolls off the tongue, that name, that work by binding to the kinase domain of IRE1, and by doing so, they prevent the RNase activity. In terms of PERK inhibitors, there's the Amgen44, which we use in the lab, and then the GSK inhibitors. Although the GSK inhibitors have been shown to have off-target effects, they're actually very good RIC1 kinase inhibitors. And then for ATF6, we've got the SEPN family, which was reported a number of years from the Walter group. So in terms of using these inhibitors in preclinical models, most work has been carried out with IRE1 inhibitors, and in particular the MKC family. So in 2018, there was 2 papers which demonstrated that the IRE1 inhibitor MKC8866 could show good efficacy in preclinical models of triple-negative breast cancer. The first of these papers was from Ji Chen's group in Baylor, and the second paper was from the Somali group in NUI Galway, and I was lucky enough to contribute to that study. And both of these studies really showed that MKC8866 when combined with chemotherapeutics such as docetaxel and paclitaxel, really increased the ability of those chemotherapeutics to reduce tumor growth. And then in 2019, there was a study also published in Nature Communications by Shen et al., in which they showed that MKC could also reduce the growth of xenograft models of prostate cancer. So in 2019, MKC 8866, which is now called ORIN 1001, moved into phase 1 clinical trials. And this update was published in 2021, which shows that it's now moved from phase 1 to phase 2 clinical trials, where it's been tested in advanced breast cancer in combination with the chemotherapeutic Abraxane, which is just essentially another form of paclitaxel. So we're at a really, really exciting crossroads in the unfolded protein response research field. So over the past 30 years, there's been so much intense research to understand the signaling pathway, to figure out the different components of it. We now know the role that it plays in cell physiology, and we also know that when dysregulated, it contributes to multiple diseases. We have inhibitors through which we can target specific control points in this pathway, and now these inhibitors are being used in phase 2 clinical trials. So as exciting as this is, and believe me, it is so exciting to see the field move in this direction, it should also cause us to just pause for a moment and ask, what don't we know about the unfolded protein response? What assumptions are we making? So I presented the UPR to you today as 3 parallel signaling pathways that are distinct but complementary. But is that really the case? And the answer there is, to be honest, it's never really been tested robustly. And if we are going to use inhibitors to switch off certain arms of this pathway, it's probably a good idea that we understand, do they only impact that one pathway, or do they impact multiple pathways? So really, if we switch off IRE1, do PERK and ATF6 signaling pathways still occur as normal, or are they impacted in any way? So in January 2020, when my first grad student joined the lab, we wanted to ask a relatively simple question. And that question was, do IRE1 inhibitors impact ATF6 or PERK signaling? So Gideon, who I think is on the call today, took MDA MB231 cells, which are a human triple-negative breast cancer cell line. And he treated these cells with thapsigargan, which is a chemical inducer of ER stress. And thapsigargan works by depleting calcium levels, and that is what induces a breakdown in protein folding, leading to the accumulation of unfolded proteins and the induction of ER stress. So when Gideon looked at ATF6 signaling, he saw that quite rapidly following thapsigargan treatment, you see this appearance of nuclear ATF6, indicating that ATF6 is being cleaved. And as we progress through our time course from kind of early stage acute ER stress more towards a chronic model of ER stress, we see ATF6 processing dissipates, and we start to see the accumulation again of full-length ATF6. In terms of IRE1 signaling, we tend to monitor XBP1 splicing as a readout of IRE1 signaling. And we could see that XBP1 was spliced from 3 hours, and we could still detect it right up to 24 hours. And when Gideon combined thapsigargan with the IRE1 inhibitor MKC8866, we saw a disappearance or a reduction in our XBP1S, suggesting or indicating that IRE1 is switched off. And when we looked at ATF6 processing, we really saw no effect on it. So it seemed that blocking IRE1 signaling with MKC was not attenuating or impacting on ATF6 signaling. So our next question was, did it impact the PERK pathway? And of course, I'm going to show you that it did, or else it'd be a very short talk today. So what Gideon found was, whenever we treat with thapsigargan, by 6 hours, we see our switch on in IRE1 signaling, shown here by splicing of XBP1. And we also see, when we look at PERK, this upshift in PERK. So if you remember, I told you that PERK trans-autophosphorylates during its activation, and you can detect this by looking for an upshift in your gel. So this is the Western blot. So we could see that PERC was clearly upshifted, and it seemed potentially to be induced. When we looked at our combination with thapsigargan and IRE1 inhibitor, we switch off IRE1 clearly through our whole time course. And when we looked at PERC, we started to notice that PERC was still upshifted, so it was still activated, but it seemed like there might be less PERC protein. Now, this wasn't just as apparent at the early time points, but when we moved to more the chronic UPR activation, we could very clearly see that when we shut down IRE1 signaling, we're leading to a reduction in PERC protein expression. So at the time when we got this result, we did ask, is this something that's just specific to thapsigargan treatment? So Gideon repeated the experiment using tunicamycin and brufeldin A, and there are two other chemical inducers of ER stress. He got exactly the same result. And then we also tried a physiological inducer of ER stress, that being serum deprivation. And again, we found that when we combined IRE1 inhibitor with our ER stressor at the later time points indicative of chronic UPR activation, we saw this disappearance in PERC protein. So our next question was, well, is this something that occurs in general, or is it just some weird artifact of MDA MB231 cells? So to answer this, Gideon took MCF10A cells and 4T1 cells. So MCF10A are a human non-tumorigenic breast epithelial cell line. And when he treated these cells with thapsigargan, we saw again this upshift and actually quite a big induction in PERC protein expression. We saw a switch on in IRE1 activity as shown by XPP1S. And when we combined with our IRE1 inhibitor, we shut down IRE1 signaling and we saw this decrease in PERC protein expression. And very similar results in 4T1 cells. Thapsigargan treatment increases or causes an upshift in PERC, indicating its activation. We see an activation of IRE1 signaling, and when we combine with our IRE1 inhibitor, we shut down IRE1 and we get less PERC protein present. We've also done this in lots of other cell lines that are not breast-derived, and every cell line that we've tested, we see exactly the same result. So this really tells us that IRE1 signaling is somehow contributing to PERC, or to helping to sustain PERC protein expression, and in particular during chronic UPR activation. So we knew this wasn't specific to one ER stress stimulus. It seemed to occur in multiple cell lines. So the next question was, is this something that's just specific to MKC 8866? So to answer this, Gideon went back and he repeated his experiment, only this time, in addition to using MKC, he also incorporated another RNase inhibitor of IRE1 for MUETC, and also a kinase inhibitor, Kyra6. And when he repeated the experiment, again, with thapsigargan treatment, he could see a switch on in IRE1 activity, an upshift in PERC indicating its activation. When he combined with MKC with 4-MUETC or Kyra6, we switch off IRE1. And when we look at PERC, we see this decrease in PERC protein expression. So this really told us that what we were seeing wasn't specific to MKC, but appeared to be general of multiple inhibitors of IRE1. Now, when you work with inhibitors, you should always be aware of the potential for off-target effects. It may be unusual that 3 different inhibitors have the same off-target effect, but we did want to approach this from a different viewpoint. And to do that, we decided to use a genetic approach. So we were lucky enough to obtain some IRE1 knockout MDA and MB231 cells. So we repeated our experiment in the scramble controls and in the IRE1 knockout cells. And what we found was in our scramble controls, as we had seen in our wild type cells, we get this upshift in PERC following thapsigargan treatments. And we see a switch on in IRE1. So it's just this little band up here. Switch on in IRE1 is shown by XPP1S. XPP1S antibodies aren't great. And the one that we use has this non-specific band that just runs under the XPP1S. So sometimes the blots can be a little bit difficult to get. But what we found was when we treated with MKC, we switched off IRE1, and we also saw this decrease in PERC protein expression in the scrambled controls. And this was very similar to what we'd seen in the wild type cells. But when we looked at the IRE1 knockouts, what we could see was that thapsigargan was still causing this activation of PERC, as shown by the upshift. but the level of PERK expression was much diminished to what you see in the scrambled controls. And when we added MKC 8866 in combination with thapsigargan to these cells, we didn't change the results in any way. The PERK protein expression was very similar to thapsigargan alone. And of course, this was reassuring to see because MKC works by blocking IRE1 RNase activity. And as you can see here, we have no IRE1 in our knockout cells. So this really suggested and kind of cemented that IRE1 signaling promotes PERK expression during prolonged UPR activation. So the next question is, well, what is the mechanism? Is it transcriptional, post-transcriptional, post-translational? So to answer this, the easiest place to start was to look at PERK transcript. So Gideon carried out qPCRs where he took MDA and B231 cells and he treated them with thapsigargan for 6 hours, 18 hours, and 36 hours. And when we looked at PERK transcript, what we saw was there was this very early increase in PERK transcript by 6 hours thapsigargan treatment. And it was sustained throughout the time course, although it does start to dissipate as you move from an acute into a chronic UPR activation. And when we combine with our IRE1 inhibitor MKC, we see that this increase in PERK transcript is reduced or lowered at all time points. So this really suggested to us that IRE1 through transcriptional upregulation of PERK was helping to sustain PERK expression. Okay, so how is IRE1 doing this? So if you remember, I told you that IRE1 RNase activity has 2 signaling outputs, IRE1 XBP1S and IRE1 RID. So whenever we knock out IRE1 or we use IRE1 inhibitors, we're taking out all of this downstream signaling from the RNase domain. So we have no IRE1 RID and no IRE1 XBP1S. So this presents a little bit of a challenge if you want to determine or differentiate between these 2 signaling pathways. But one way you can do it is by knocking out XBP1. So if you take XBP1 knockout cells, they still have IRE1. So they're still going to have IRE1 RID signaling, but there's no XBP1 to splice. So there's no IRE1 XBP1S signaling pathway. So we obtained XBP1 knockout cells and their scrambled controls. And what we found was when we treated the scrambled control cells with thapsigargan, again, we see our increase in PERK transcript, and this is prevented or reduced when we combine with IRE1 inhibitor. But when we look in our XBP1 knockout cells, we don't see this increase in PERK transcript following thapsigargan treatment. So this would suggest to us that IRE1 XBP1S signaling is playing a role in this. An important control that we did here was to combine thapsigargan with IRE1 inhibitor in these XBP1 knockout cells. And this was important because if you remember, I told you these cells still have RID activity. So if RID contributes in any way, by comparing these 2 treatments, we should see it. And as you can see, they're virtually identical. So this really indicated that what we're seeing is an IRE1 XBP1S dependent signaling. So in addition to looking at transcript, we also looked at protein levels. And as we had kind of seen before, when we treat our scrambled controls with thapsigargan, we see an upshift and an induction in PERK protein. Combined with IRE1 inhibitor, we see a reduction in PERK. When we look at our XBP1 knockout cells, so you can see these cells have no XBP1S, they still have IRE1. And when we look at PERK, we see this upshift in PERK following thapsigargan treatment, but the level of PERK protein expression is much, much less than what you see in the scrambled controls. And when we add MKC in combination with thapsigargan, we don't change this in any way. So these results really indicate that IRE1 XBP1S signaling helps to sustain PERK expression during long-term UPR activation. So the next question that we had was, if IRE1 inhibition is reducing PERK protein expression, is this impacting on downstream PERK signaling? And the way we chose to assess this was by looking at phosphorylation of EIF2 alpha. So again, if you remember at the start, I told you PERK phosphorylates EIF2 alpha. So we went to our scrambled and our XBP1 knockout cells, and they were treated with a time course of thapsigargan. So in the scrambled controls, you see an upshift and very clear maintenance of PERK protein expression. In our XBP1 knockouts, we still see this upshift, but we've got much less PERK protein present. And when we looked at phosphorylation of eIF2 alpha in the scrambled controls, it's very clear that PERK is phosphorylating eIF2 alpha. And in the XBP1 knockouts, it's still there, but it is much reduced. So it seems that by lowering PERK protein expression, we may be impacting on downstream signaling. So just to summarize, our results really show that IRE1 through XBP1S signaling during chronic stress helps to maintain expression of PERK protein, and this may help to maintain downstream signaling. When we use IRE1 inhibitors, we prevent RNase activity. So this prevents this upregulation in PERK and leading to reduced PERK expression and reduced downstream signaling. So our next question is, what are the functional consequences of this? And due to time limitations today, I'm not going to get into the data we have in that, but I will say that it does appear that there may be an impact on cell death kinetics. So to conclude, our results indicate that IRE1 signaling boosts PERK expression during chronic ER stress. It suggests that crosstalk does exist between UPR mediators, and it also should suggest that when we're using IRE1 inhibitors and potentially IRE1 activators, that we should be open to the fact that they could be impacting multiple arms of the UPR and not just one, as is or as tends to be assumed. And finally, we know a lot about the UPR, but this work really shows us that there's still a lot to learn about the intricacies of this signaling pathway. And just to finish, I think Gideon needs to be acknowledged here. He started his master's in January 2020 and is now a PhD student. And as you know, trying to start your research over the past 4 years has not been an easy task. So he's done a wonderful job. Rosamond, who is a summer student who was with us last year and helped set up the conditions for physiological induction of the UPR, and hopefully he'll be back in the lab in another few weeks. Babushin, who is a master's student in the lab, who's working more so on IRE1 within the context of how it promotes triple negative breast cancer. And then Wafa, who was a research associate in the group, who really was helpful in getting the lab set up and kicking off some of these projects. And of course, acknowledge our funders, including NSERC and Canada Research Chairs and collaborators who very kindly provided knockout cells. And I will be happy to take any questions now.
All right, thank you so much for that, Dr. Luke. That was a great talk. So if anyone has any questions, feel free to raise your hand or drop them in the chat this time. Go ahead, Matt.
That was a really nice talk. Very, very impressive work and a cool mechanism. I had a question about some of the data with the eIF1 alpha phosphorylation data, and maybe this is just a Western blot artifact, but when you had the XBP1 knockout cells at the 36-hour time point, the very last time point, it looked like it was a little bit higher. Do you think that's real, like there's another kinase or something, or is that just an artifact?
It also seems to be the way in the scrambled controls as well. It's like it goes up again at 36 hours. So yeah, we have noticed that, and we don't have an answer for it. I mean, obviously PERK is not the only kinase that can phosphorylate eIF2 alpha. There's 3 other kinases. So I'm not sure if something else is being triggered at that point, but yeah, well spotted.
Interesting.
Right, I'm not seeing any other questions. Oh, one more. Go ahead, Michael.
Hi, can you hear me? Yes.
Yep.
Yeah, great talk. I really enjoyed it. I was wondering, I mean, it seems maybe these 3 pathways of UPR are portrayed as being non-redundant, but they maybe they seem like they are connected and, you know, they're not functionally separate. I'm wondering, is it possible to activate the perk separately from the IRE and then test that?
Yeah,
Oh.
so sorry, I think the question
No,
was
I'm
breaking
sorry.
a bit.
No,
Yeah.
go ahead.
So yeah, there are small molecule activators of PERK, but one way you could just activate the PERK pathway is just to overexpress PERK because it dimerizes and kind of auto activates. So if you overexpress it to a high enough level, and that is something that we're starting to try and do in the lab, to develop systems to try and activate these signaling mediators without the need for using chemicals. Because whenever we use chemical inducers of ER stress or physiological inducers, you're not just activating ER stress, you're triggering a lot of other events within the cell. So if we could kind of take that out of it and have a way to just control IRE1 expression and a way to control PERK expression, it may allow us to start looking at the dynamics. Because as you said, these are often portrayed as three separate signaling pathways that work collectively, but actually the UPR is a really dynamic process. So certain arms will switch on fast, some will be maintained, others will switch off.
Yeah, thank you.
Okay, so with that, I don't see any other questions. We'd just like to thank everyone for joining us for this session. And just a reminder that we do have another session coming up in May, where we will hear from Louis from the University of British Columbia, as well as Dr. Afar from the University of Montreal. So if you'd like to join us, please feel free to sign up and share this link with your labs and departments. And with that, thank you for joining and have a great day.
Thank you. Bye.